Welcome. In this session, I'm going to begin a new chapter, Biotechnology Principles and Processes. I hope you still remember the molecular basis of inheritance, in which we discussed about the DNA molecule, okay, in detail. And now in this chapter also, I'm going to deal with that DNA molecule, how I'm going to manipulate the DNA molecule and use it for various beneficiary purposes of human beings. Okay, so now let's see what actually this biotechnology is. So this biotechnology is a combination of biology and technology. So it's a combination of biology and technology. Then what this biotechnology is going to deal with? So it's going to deal with the live organisms. So we are going to use these live organisms and also enzymes from organisms. So the live organisms and enzymes from the organisms are used to produce products and processes that will be useful to humans. Okay, so what are the products and processes useful to humans? Like we say, making of curd, so making curd, then bread, wine. Okay, so these are all, of course, we know from several thousand years ago. So we started making these curds, bread, wine. So these are also processes of biotechnology. But now, in the modern years, the way of biotechnology, so the way we look at the biotechnology is changed. So in this case, in the modern view, the biotechnology, the biotechnology, so I can say in terms of modern biotechnology. So in this case, we especially deal with something called GMOs. So which means these are genetically modified organisms. So these are genetically modified organisms. Right, we know that we have come to know the structure of DNA, and we also have come to know that where the genes are present, the locations of the gene and everything. Even after the Human Genome Project, we started sequencing many other organisms, and we have complete information of the genes in Drosophila, in many other plants, including human beings. Now, okay what we are going to do is that, say suppose in this DNA molecule, so there is a particular gene at particular position. Now I'm going to cut this gene. So this is, say suppose, the gene X. I'm going to cut this gene from the DNA molecule of an organism, and then I'm going to put that into another organism. So this gene X, what I'm going to do, so I'm going to cut here, and insert this gene X into this point. So this is, say, DNA of an organism. DNA of an organism says it is A, and this is the DNA of an organism B. So that is the DNA of an organism B. So here the gene X, something, gene, so which I'm going to cut it from this A organism, and I'm going to insert into the DNA of organism B. So at this point, now this organism which I said B is going to become something called GMO because I have modified its genetic information. So it's called the genetically modified organisms. So in the modern terms, the biotechnology will be usually dealing with these genetically modified organisms. Okay, so to produce products, okay, in large scale. In large scale, say suppose I give you an example that insulin that is produced, okay, by the pancreas in human beings, okay, the gene that is coding for insulin is removed, okay, from the DNA of humans, and then that we have inserted into a bacterium. So we kept it into the DNA of the bacterium and made the bacterium to produce this insulin. Okay, now that bacteria is what we call the genetically modified organism, and we are going to use that bacteria, or grow that bacteria in large scale to produce insulin in large scale, so that it can help so many diabetic patients all around the world. Okay, so in modern sense, the biotechnology will deal especially with these genetically modified organisms, which we are going to modify its genetic material by taking out some genes from another organism and inserting into its genetic material. Okay, so that's about the genetically modified organisms. And let's see some other examples of this modern biotechnology. So here, even in vitro fertilization, in vitro fertilization is the one which also comes under this modern biotechnology. So in vitro fertilization, which means the fertilization that we do it in the lab in a test tube. Okay, so this is in vitro, which means outside the body. We have two words, in vivo and in vitro. So in vivo, which means inside, which is a natural, if I consider this fertilization, okay, within the female body the fertilization takes place. So in vitro means I'm going to collect the sperm and egg, okay, and then I'm going to fertilize in, or allow them to come together. Okay, so here, say this is the egg and the sperm, right, in a test tube. So this is in vitro fertilization here. So that's what we have studied in reproductive health. Okay, so under assisted reproductive technologies, those couples who have some issues and cannot conceive, we have gone with that reproductive assisted reproductive technologies, right? So that's an in vitro fertilization. This can also come under the biotechnology, right? Of course, this in vitro fertilization, the result is the test tube baby. Okay, so this is also considered under biotechnology here. And then synthesizing a gene. So because we have come to know what is a gene, what it is made up of. So a gene is simply made up of a sequence of nitrogen bases. We know those nitrogen bases, adenine, guanine, thymine. Okay, right. So these nitrogen bases, when you keep it in a order, of course each gene, I told you in the previous chapter, that made up of a particular sequence of these nitrogen bases. Okay, so each gene has its own sequence of nitrogen bases. Now if we know that sequences, we can even synthesize that gene in the laboratory, okay, by using these nitrogen bases and what are other components that are required to make it. Okay, so that's even this synthesizing a gene. So synthesizing a gene is also the part of this biotechnology, right? And then even developing DNA vaccines. So even developing these DNA vaccines, okay, and even correcting a defective gene. So correcting a defective gene. Okay, so this is actually called gene therapy, which I'll discuss in detail in the next chapter, biotechnology and its applications. Okay, so correcting a defective gene, so where we can replace a gene that is non-functional with a gene that is functional. That what we call it gene therapy, and is also part of this modern biotechnology, right? So all these are, the various uh, techniques that all come under modern biotechnology, whereas making, the curd, bread, wine and all, they come under the classic biotechnology, means it's an older biotechnology, whereas the especially dealing with genetically modified organisms and even developing test tube babies, then synthesizing gene DNA vaccines and correcting a defective genes, all will be a part of modern biotechnology. So now let's see what actually or how we can define the biotechnology. So this biotechnology is the branch of biology that will deal with the use of microbial organisms. So it's a branch of biology that deals with the use of microbial organisms, so like bacteria and all, right, and their products and their, okay, products for the benefit of, for the benefit of human beings. So of course this is a older definition. A newer definition is given by EFB. Okay, so EFB, they have given a newer definition. So this stands for European Federation of Biotechnology. So European Federation of Biotechnology, they have given a newer definition for, okay, biotechnology. So what is that a new definition for biotechnology? So here the definition goes something like this. So the integration, okay, so the integration of natural science and organisms, the integration of natural science and organisms, right, cells, okay, so the organism cells, then the parts thereof, okay, so that parts also we are going to include in this, and then, okay, molecular analogs. So even we are going to include this molecular, okay, analogs, okay, for products and services. So this is the new definition, the integration of natural and organisms, so their cells, their parts, and even the molecular analogs, which we will discuss, okay, in the coming sessions, okay, for products and services. So this is the new definition that is given by EFP. So whereas the older definition, so it is just like using microbial organisms and their products for the benefit of human beings. Now it is not just using the microbial organisms, okay, So we are going to use the parts, okay, the molecular analogs and whatnot for the products and services. So this is the modern, okay, biotechnology definition. Now let's look at uh, this biotechnology, the modern biotechnology. It has two main branches. So the modern biotechnology, it has two main branches. So one, it is genetic engineering, and another one is bioprocess engineering. So it's called genetic engineering and bioprocess engineering. So what is this genetic engineering? So it is going to deal especially with the techniques that are going to alter the chemistry of genetic material. So genetic engineering, it deals with techniques to alter the chemistry of genetic material. Okay, so it's going to alter the chemistry of genetic material, and of course that will be by introducing the foreign DNA. So just now I have mentioned over here, I have taken the DNA molecule of an organism A, and I have taken the organism B. So what I did is I have taken a portion of the DNA, so that I have taken it as gene X. So this gene X will be the foreign DNA. It will be the foreign DNA, which I am going to place it in the DNA of B organism. Okay, so this one I am going to write insert into that B organism. So this part of DNA, that which I have removed it from the DNA of an organism A and introducing into this organism B, that will be called the foreign DNA. So the genetic engineering will deal with, okay, how to cut that gene, right, and then insert that gene into B. So we cannot do it manually by, we cannot see the DNA molecule, we cannot cut it with the scissor, and then just how we paste the paper, cut a paper from one and then paste it on another. That is not possible, okay, in case of DNA molecule, because we will not be able to see with our eyes the DNA molecule. So there are some special enzymes, and with that I'm going to cut this gene X from the DNA of an organism, and then I'm going to place it into another organism's DNA. So that's what the genetic engineering is going to deal with, all those. Okay, so introducing the foreign DNA into the host organism. So into which the foreign DNA I am introducing, that will become the host. So this B organism in this case is host. If I am taking the gene from the B and introducing into A, then the A will become host. Okay, right. So in this example I have given the B as the host here. So introducing that foreign DNA into an, okay, organism, so that is called the host organism here. So with this, what happens, there will be a change in the phenotype. So there will be change in the phenotype of the host organism, because I have introduced, okay, some genes into the host organism B, and obviously there will be a phenotypic change in that host organism. So that's all with this genetic engineering, which will deal with okay, altering the DNA molecule. Okay, then coming to this bioprocesses engineering. So in the bioprocess engineering, so what we see here, it will include the maintenance of a sterile environment. So in this case, it is maintenance of sterile environment. So which means it is a microbial free environment. Say suppose if I'm going to conduct some biotechnological processes in this room, then definitely this room should be free from microbes. So there are so many microbes that will be present around me, okay, and they will be constantly trying even to enter into my body. And so we don't want all these microorganisms wherever I'm going to conduct these biotechnology technique. So the room should be very much clean. That is should be sterile. So the here sterile environment, which means it is a microbial free environment. Okay, so such sterile environment will help only the growth of the organism that we want it to grow. Say suppose I have chosen a particular microorganism, say E. coli. I want to grow only E. coli. But if this room is not sterile, and if I take a nutrient medium and then put E. coli on that, then what happens is, so you can take, usually in the labs we grow by taking a nutrient medium and placing that bacteria, like say suppose E. coli, and this E. coli will start multiplying in this nutrient medium, and it starts becoming a colony. It becomes a colony. But if the room is not sterile, other microbes will also settle on this nutrient medium, and they also start multiplying and becoming more number. So then we will not be able to perform our experiments properly. So we require a sterile environment for growing the organisms of our choice. Okay, so this allows the growth of only desired microbe, which means only the microbe which I want it to grow, I can grow it in the sterile environments here. Okay, and of course I'm going to grow that microbe in large quantities, so that whatever the product is, say suppose what I have did, as I mentioned earlier, the insulin gene that has been isolated from the human DNA, now we have placed into a bacterium, and allow this bacterium to multiply and become in large number. Then only we will be able to get enough quantity of insulin. Okay, so I'm going to grow in the large scale. So this desired microbe is grown in large scale. So how we are going to grow that desired microbe in large scale, what are the instruments that we are going to use, all those things I'm going to discuss in our coming session. So just this is I'm just introducing that the branches of this modern biotechnology. First we will see about that genetic engineering, how we are going to alter that DNA molecule. Then we will see about that bioprocessing engineering also. Right, so now let's see the genetic engineering. Genetic engineering is also called rDNA technology, also called rDNA technology. So here the R stands for recombinant. So in this case, the R stands for recombinant, okay, technology, or you can also call it simply as the gene technology. So what is this R DNA? So I'm going to take again the same example. Okay, say this is the DNA of organism A, and this is the DNA of organism B. So I said that I have taken or selected something called a gene X from this organism A, and I'm going to place it to the organism B, right? So here, now when I place this into organism B, so you can see here a small portion I have, right? So this is the DNA from this point, okay, from organism A. Now as I have changed, okay, or altered the DNA molecule of organism B, this is what I call the rDNA molecule. So this is called the rDNA molecule, that is the recombinant DNA molecule, okay, right? So let's see in this genetic engineering, so the point is that how I'm going to select this gene, and how I'm going to cut this gene, and then how I'm going to place it, and then okay, paste it in that another DNA molecule. So for all this it requires tools. So see, under this genetic engineering we have, okay, mainly three steps. That is, one, creation of rDNA. So creation of rDNA, I hope you understood what is that creation of rDNA. Then the second one will be the gene cloning, and the third one will be the, okay, the gene transfer. So now, what is this gene cloning and gene transfer? All that I'm going to discuss in detail. Okay, so here the steps of genetic engineering: first creation of rDNA, then gene cloning, and then the gene transfer. Let's see one by one. So first we'll see here how we are going to create this rDNA. So that's tools used in rDNA technology. So what are all the tools that are required? So I told you we need to isolate the gene what we want, and then we need to of course cut it from that organism, the source organism, and then we need to put it into the host organism. So for that we require some tools. So those tools, important tools which are used in genetic engineering, will include something called the restriction endonucleases. So they are restriction endonucleases. Then we require something called the cloning vectors, and then we also require DNA ligases, and then the polymerases. So we require the enzymes, okay, so like these polymerases, okay, and all these and of course the host organism. So here especially I'm going to do or explain the first three, because these are the main tools which are used in making rDNA. So restriction endonucleases, cloning vectors, and the ligases, and what's their function and how they are help in creating the rDNA. So first we'll see about these nucleases. So these nucleases are enzymes, and are of two types. So they are called exonucleases and another one called endonucleases. So whatever the nucleus, the nucleases are the enzymes that are going to break the phosphodiester bonds. So they break phosphodiester bonds. Okay, so if you see, this is the DNA molecule. Right, so we know that the phosphodiester bonds will be present here. Okay, so these are hydrogen bonds, and in between this sugar and the nitrogen base, we have N-glycosidic bond. So the point here, it's going to break these phosphodiester bonds here, and thereby separating the DNA. So if I want this piece, so say suppose this is a gene, so I'm going to cut this phosphodiester bonds, and I can isolate this fragment of DNA. So nucleases will break the phosphodiester bonds, not hydrogen bonds or N-glycosidic bonds. Now, what is the function of these exonucleases? So these exonucleases, usually they remove nucleotides from ends of DNA. So they are going to remove the nucleotides from ends of DNA. So when we see the nucleotides, so this is the nucleotides, and what the exonucleases are going to do, so 5' and 3' say, and they are going to remove these this is a nucleotide. So say suppose this is one nucleotide, this is the second nucleotide, and it goes on nucleotide by nucleotide. It's going to remove nucleotide by nucleotide. So first it's going to separate this nucleotide. So you can see the nucleotide is separated. So one by one it's going to remove from the ends. So it is not going to cut the DNA at the center. That is the function of endonuclease. So endonuclease will cut the DNA molecule. Okay, so it's going to cut the DNA molecule, or I say cut DNA molecule, or cut within the DNA molecule. So they are going to cut the DNA molecule within. So that's if you see here, this is a AT. Okay, so this combination, so you can see that in this case it's going to cut somewhere here. Okay, of course that I will tell you that how, where it's going to cut. Okay, so there is a lot of process that we need to know how these endonucleases actually act. So exonucleases, they just remove these nucleotides from the ends of DNA, whereas the endonucleases, we are going to cut within the DNA molecule. So we don't want exonucleases. So here, for making the rDNA, the endonucleases are most important, because if I want a particular segment, so from this point to this point I want, then I can use these endonucleases and cut that fragment of DNA within that DNA molecule, right? So one example of these endonucleases is that the restriction endonucleases, right? In short, it is called REN. I hope you remember this endonuclease word when I discussed about DNA fingerprinting. So when I discussed about DNA fingerprinting, there we have seen, once we have taken isolated the DNA, we need to cut the DNA into fragments, and that fragmentation we have done it by using this restriction endonucleases. So we are not we will be not using these exonucleases. So here our point, which is very important in making our DNA, is endonucleases. So as they are going to cut the DNA molecule within the DNA, right? And in the year 1963, so in 1963, okay, scientists found two enzymes that actually responsible for restricting the growth of a bacteriophage in E. coli. So we know bacteriophage is a phage, so which means it's a virus infecting E. coli. So here they found that, okay, so E. coli is producing two enzymes. It's producing two enzymes to restrict the growth of bacteriophage. So it is producing two enzymes to restrict the growth of bacteriophage. So one of that enzyme, so one enzyme, it added, so say suppose this is the first enzyme, so the first enzyme, so this added methyl groups, is added methyl groups, okay. And then the second one enzyme, so it cut DNA. The second enzyme is used to cut the DNA. Okay, now one question: if this E. coli bacteria is able to produce an enzyme that is able to cut the DNA of, say, this bacteriophage, why not, okay, this enzyme is able to cut the bacterial own DNA? You find out the answer, okay, and we'll see that answer in our coming sessions, or uh, you find out the answer what actually the reason for not cutting the E. coli's DNA by this enzyme. So of course this enzyme is what I call the restriction endonucleases. So the restriction endonuclease enzymes are produced by the organism itself. Say suppose in this case E. coli. So this E. coli is producing this restriction endonuclease and is able to cut this, the bacteriophage DNA, right? So that's what my question is: if it is able to cut the bacteriophage DNA, of course, is the bacteriophage DNA different from the bacterial DNA? That's one question, okay. And another question is that how this RNA is able to cut only that bacteriophage DNA and why not the E. coli's DNA? Okay, so find out those answers. So right, so this is the restriction endonuclease, which they cut the DNA, which I have already mentioned. Now the first restriction endonuclease that was discovered, so first REN, discovered is Hind2. So the first enzyme discovered, that is REN discovered, is Hind2. Of course, there is a nomenclature that we give to the restriction endonucleases that we discovered, okay, from these organisms, okay. So different groups of organisms will produce, okay, different restriction endonucleases, right? And we have one set of rules to give names to those restriction endonucleases, which I will be discussing, okay, in coming sessions. So here the Hind2 is the first restriction endonuclease, okay. And these restriction endonucleases, say suppose this the first restriction endonuclease Hind2, so it used to cut DNA only at specific locations. So it cuts the DNA only at specific locations. It's not going to cut the DNA, okay, at random sites. And now the point at which the restriction endonuclease cuts is what we call a recognition site. So the point at which a restriction endonuclease cuts DNA, so that is called the recognition site. So it means that the restriction endonuclease recognizes, okay, the site, okay. Of course, each restriction endonuclease has its own recognition site, okay. So that particular site, only at that particular site only the restriction endonuclease will act and cut the DNA. The point at which the restriction endonuclease cuts the DNA is what we call the recognition site. Right now, of course, in the beginning we know only a few restriction endonucleases, but now what we have come to know. So later we started discovering a lot many restriction endonucleases. At present, we have more than 900. So more than 900 restriction endonucleases discovered. So we have come to know more than, okay, 900 restriction endonucleases discovered, and that too from, okay, from 230 strains. So from 230 strains of bacteria. Okay, right. Fine. So this with this I'll end today's session. So in the next session, I'm going to discuss the nomenclature of these restriction endonucleases, and also we will say about what is that gene cloning, okay, and gene transfer, and how I'm going to make the host to express, right, whatever the gene that I'm going to introduce into it.